Hey, and welcome back. To put in context what we've been talking about in terms of peptide structure and dihedral angles, all of this is connected to the central dogma of molecular biology, which is that information in biology flows from DNA to RNA and then to proteins through sequence. But as we think about proteins, within that sort of category, there's a connection between the sequence of a protein and its structure, and then a connection between its structure and its function. And we're going to be focusing in on this connection between sequence and structure. And there's a lot of factors that govern the relationship between sequence and structure. That is the order of various amino acids in a particular peptide or protein, and then what shape it adopts. And these factors can include dihedral angle preferences of specific amino acids. And they can also include various non-covalent interactions, which you learned about in 351 and we'll talk about again. And then also something called the hydrophobic effect, which I think you also learned about in hydrophobic effect, which you also learned about in 351. And it's the reason, for example, why oil and water don't mix. So last time we spent a while talking about defining dihedral angles for amino acids in peptides and proteins. Rotation around this nitrogen to alpha carbon bond is given the name angle phi, and rotation around the bond between alpha carbon and carbonyl carbon is given the name psi. And these phi-psi angles are important because they define the secondary structure of a peptide or protein. So we went through 3 different amino acids in this helix bundle, where you've got 3 different alpha helices packing against each other. And we looked at each dihedral angle. Again, here is phi, which you get by measuring the angle between the carbonyl carbons while looking down the nitrogen to alpha carbon bond. And then this angle psi, which you get by measuring the angle between nitrogens while looking down the alpha carbon to carbonyl bond. And we measured those dihedral angles for this string of 3 amino acids within this peptide, and these are the numbers we came up with. And those numbers define alpha helical secondary structure. Now, one amino acid at one isolated place in a protein for sure can adopt this dihedral angle, but if you have a string of them consecutively that adopt those angles, you're going to get an alpha helix. So secondary structure is really defined and described by these phi and psi angles. And then we looked at this Ramachandran plot where each of these dots represents an individual amino acid in a protein structure of the thousands and thousands of examples that there are. And we're plotting the phi-psi values there. And you can see they cluster in specific areas of this plot. Mostly negative values of phi, and then positive and negative values of psi are tolerated. If you have phi-psi angles in this area, those tend to be consistent with helical secondary structure. Not every example that's in here is within an alpha helix, but if you had a string of consecutive residues with those dihedral angles, it would for sure be in an alpha helix. And then up here you have dihedral angles that are consistent with the beta pleated sheet. Also shown, not particularly illustrated here, are what are called polyproline helices, which, if you've ever heard of collagen, it adopts dihedral amino acids in collagen adopts dihedral angles sort of in this area of the Ramachandran plot. One thing to note is that most of the examples of amino acids we have in proteins have L stereochemistry. So you might imagine some dihedral angles to be preferred over others. And you can see that all sides of the Ramachandran plot are not populated evenly. You have more examples where phi is negative than where phi is positive. There are some examples here, but just as a point here represents alpha helical secondary structure because you have phi psi values of around minus 57 and minus 47. So over here, if you have plus 57 and plus 47, you're also in an alpha helical conformation, but it's left-handed instead of right-handed. And we talked about handedness looking at this alpha helix. If you point your thumb in the direction of the C-terminus of the helix and then wrap your fingers around, if it fits your right hand, that's a right-handed helix. All right, and the last thing I wanted to point out is that side chain structure, this R group here and then here, clearly influence the phi and psi angles because the R group is going to determine whether it's better for the carbonyl in the front to be closer to the carbonyl in the back or closer to the side chain, and it's going to determine whether the nitrogen in the back should be closer to the nitrogen in the front versus the side chain. So just through steric arguments, you could expect side chains to influence phi and psi values. Okay, so that's the brief summary. What you want to know is what's going to be on the test, and I think it's fair, since we've talked about alpha helices so much, for you to know the minus 55, minus 47 angles are consistent with alpha helical structure. Frankly, though, that's a boneheaded and easy question to ask on an exam, so I'm probably not going to ask it because all you got to do is memorize. But I will ask you to think and use the things that we've talked about to draw some conclusions. For example, I might tell you that a particular combination of phi and psi amino acid, phi and psi values is really, really good at a particular spot in a protein. And then I might give you some data about which amino acids prefer which angles, and you could suggest which would be the best at that position that we've talked about. Let me show you what I mean. Here is a plot of, here are the structures, names, structures, and pKa's and phi-psi preferences for the 20 amino acids. So I want you to memorize amino acid structures, their names, and their one-letter codes. You could memorize their three-letter codes, but if you know the names, the three-letter code is basically obvious. So names, one-letter codes, and structures. They all have the same backbone structure, it's just the side chain that varies. I'm not going to bother writing all of them down one by one here because you can memorize on your own time. It's not like it's going to be that effective if we try to do it together. But I've grouped the 20 amino acids into a few categories that sort of broadly give you a sense for what those side chains might do. And as we talk about each one, I'll show you a Ramachandran plot for just that amino acid in all proteins, so you can see what kinds of secondary structures or what kinds of phi-psi angles are adopted by these amino acids. So in green, you have a group of nonpolar amino acids that are mostly sp3 hybridized saturated carbons, right? In purple, you have amino acids that are nonpolar but are aromatic. And these are interesting because they are sort of greasy and nonpolar, but they have some other things they can do too. In blue are the side chains that are cationic or can be cationic at physiological pH. This is really important, and you've seen this before, I think, on your first exam when I asked you to use pKa information to predict what groups would be positively charged versus not. Positive charges can be really important in protein structure. You can have electrostatic interactions that either attract two groups together or make them repel. In red are the negatively charged side chains. In yellow are the side chains that are polar but neutral. And then in pink are the weirdos. And these are the ones that are very different from the other 18 amino acids, and we'll talk about why. So let's start with the nonpolar amino acids. You have alanine, and again, this is going to be in your notes. You're going to have to do whatever it takes to memorize these. You don't have to memorize the Ramachandran plot. You should be able to interpret it if I gave you data. And of course, I'm not going to expect you to draw one of these from memory. Alanine is just a methyl group. The Ramachandran plot here is a heat map where more intense colors of red mean more examples. So places where the plot is intensely red suggests that there are a lot of examples of alanine with dihedral angles that are consistent with alpha helical structure. But alanine is sort of happy in a lot of different contexts, in the beta sheet context and in the polyproline helix context. So it's a fairly decent go-to amino acid. And when protein chemists want to determine whether a side chain is important or not, they will often replace it with alanine, which is just a methyl group, with the idea that alanine's happy most anywhere. And so if you want to see what difference, say, a positive charge side chain makes, you swap it out with this neutral sort of innocuous bystander amino acid to see what the major effect is. Valine is a little bit bigger. Instead of you attach two other methyl groups to the beta carbon. This is the alpha carbon of the backbone, and then side chains begin with the beta carbon. Valine's one-letter code is V. That's easy to remember because its side chain sort of has a V shape, two methyl groups. It's interesting. It's only one of the only few amino acids that has a branch point at the beta carbon. So you've got two equivalent or two gamma methyl groups. We'll call this a beta branched amino acid. And the inclusion of that branch makes a difference. Valine is happy in alpha helices. It's more happy than alanine in beta sheets, and that is because of this branching. And then it doesn't really fit well in the polyproline helix type area. So already we're seeing some differences in context, and this is one of the ways that sequence can influence structure. Leucine is like you take valine and you stick another methylene group in between the alpha carbon and the branch point. So in leucine, the beta carbon is a CH2 group. Then you have a branch point at the gamma with two methyl groups here. Different from valine, leucine's quite happy in alpha helices, a little less happy than valine in the beta strand conformation. And then isoleucine is an isomer of leucine. So that's why it's called isoleucine. I don't know why leucine got to be called leucine instead of which one's leucine and which one's the isomer. I don't know. Chicken or the egg. This one with no beta branching is leucine. If you take one of the methyl groups of leucine and move it to the beta carbon, you have isoleucine, but you've just made a stereocenter, and that stereocenter has a specific configuration that it always is in nature. So with isoleucine, you got to worry about two stereocenters. You got to get the alpha carbon stereocenter right and the side chain stereocenter, and this is its configuration. Notice that isoleucine is a lot like valine. Their Ramachandran plots are almost identical because of this beta branching. They're both decently happy in alpha helices, both quite happy in beta sheets, not so much in the polyproline area. And then finally, you've got methionine. This is kind of a weird one because instead of just being a hydrocarbon, you've got beta, gamma carbons, and then the next atom is a sulfur, followed by a CH3 group. Why does nature want to do this? Well, nature actually does a ton of other things with methionine. If you put something else here, the sulfur becomes positively charged, and the methyl group gets reactive. And so if you want to do SN2 reactions in biology, you'll often use something called S-adenosyl methionine, or SAM. As it is here, though, it's relatively nonpolar. It's comfortable in alpha helical context, a little bit less in beta strands. One challenge with methionine is sulfurs are really highly oxidizable. And so if you have a protein or a peptide with methionine in it and you leave it sitting out in air, almost inevitably you're going to oxidize that sulfur to this sulfoxide, and that can have some consequences for protein structure. All right, so those are the nonpolar ones. Let's talk about the aromatic ones. Oh, any questions you want to ask so far? Do we have to memorize everything I just said? No, but this is background to help you use information and data to draw conclusions. Yeah?
Just to clarify, these Ramachandran plots are just plots of key specific amino acids across like all proteins.
Right. It's a plot. The Ramachandran plots are plots of the phi-psi angles adopted by this amino acid methionine in all proteins. So you get to see all the different contexts that it's found in. Okay. Others? When I say secondary structure, is that understandable? I'm talking about alpha helix, beta strand. Yeah. Yes?
Little recording boxes.
The little recording box is in the way. Thank you. Always. All right. Hey, at least I pressed record this time. Go ahead.
Do the angles of like the actual side chains don't matter, or do they measure those?
Oh, that's a great question. So the side chains, you have rotatable bonds here, and they do categorize those angles. In general, the side chains are a lot more flexible than the backbone atoms are. But yes, they do. I think this bond between alpha and beta, that dihedral angle is chi1, then chi2, then chi3, and there's all kinds of definitions. And there's stuff to be learned there, but not that we're going to care about. But you can if you want to, and the people do talk about that. Did I see another hand? Yeah. Can I explain the polyproline thing? Yeah, so proline is this amino acid here. It's unlike any of the other amino acids because the nitrogen in the backbone is tied back to the side chain in a 5-membered ring. So the proline amino acid by itself is a secondary amine, whereas all the other amino acids are primary amines. And the fact that this is a 5-membered ring constrains the phi angles. There's some angles that proline just can't adopt because you start rotating around and you break the ring, right? And so relative to another amino acid, let's just look at methionine over here. Methionine adopts a lot of different negative values of phi, but proline, because of that constraint, only adopts a very narrow range of phi values. So if you put a lot of prolines together, one after the other, you tend to form a helix, but it's not the same as the alpha helix. And these helices that proline, that extended sequences of prolines form, actually tend to wrap around each other. So you get this weird triple-stranded structure, and I'm not representing it particularly well, but that's sort of the collagen triple helix. If you've ever heard about collagen, it's a structural protein, but it forms this triple helix, and it's got a lot of proline amino acids in it. So, and the dihedral angles adopted in this structure tend to show up here. There's more than one kind of polyproline helix. There's type 1 and type 2, and who knows what those mean. But it's just a different kind of structure than either the alpha helix or the beta sheet. Yeah, what else? Okay, so let's talk about the aromatic ones. Phenylalanine is like you attach a benzene ring to alanine. That's why they call it phenylalanine. Its one-letter code is F. That's easy to remember because phenylalanine. The side chain is aromatic. And one thing you need to know about aromatic side chains, if you didn't know this from 351, it's good to know it now. sp2 hybridized carbons are more electronegative than hydrogen. And what that means is that in a benzene ring, the hydrogens are electron poor and the carbons are electron rich. So even though benzene isn't polar, there's no dipole, there is something called a quadrupole. And a quadrupole is a non-uniform distribution of electron density, but not along a line, instead in space. So if you were to look at the electron density associated with this ring, you would see a lot of it hovering above or below the benzene ring. And then around the edges, you would see electron poorness. And that means that aromatic side chains can do things that just regular hydrocarbon side chains can't. For example, imagine the electron poor edge of one benzene ring pointing down and stacking on the face of the electron-rich face of another benzene ring. That's called a T-shaped benzene interaction, and it's pretty strong. Or imagine a cation of some sort, perhaps sodium or potassium, hovering over the electron-rich face of a benzene ring. That's called a cation pi interaction. And it's an attraction between the cation and the electron-rich face, and that's found all over proteins and is important, for example, in the acetylcholine receptor and neurotransmission. Tyrosine is just like phenylalanine, it's just the phenol version. You put an OH group here. The pKa of that phenol is around 10, so sometimes tyrosine side chains can be negatively charged in different situations. Notice that both these amino acids, phenylalanine and tyrosine, are comfortable in alpha helices, but also comfortable in sheets. And then finally, you have tryptophan. Tyrosine, the one-letter code is Y. That sort of makes sense because there's a Y in tyrosine. For tryptophan, the one-letter code is W, and I've been thinking about tryptophan for like 20 years, and so it just naturally is in my mind that it's W, but I remember first learning that and thinking, where the heck did the W come from? And the answer is we ran out of T and Y, and we're using P for something else. So the best they could come up with is tryptophan. So if you say tryptophan like you are either Elmer Fudd from the Looney Tunes, which I realize is a reference most of you are not going to get, or alternatively, a 3-year-old who has trouble saying his or her R's, tryptophan, and then it starts with W, okay? And if you haven't seen Elmer Fudd, it's from a simpler and less politically correct time, but he stomps around the woods chasing Bugs Bunny with his double-barrel shotgun, saying things like, "Be very quiet, we're hunting wabbits." And you should really look up the classic, the Looney Tunes classic, the Rabbit of Seville, which is Bugs Bunny and Elmer Fudd parody on the Barber of Seville. Anyway, sorry. So tryptophan does the same things as phenylalanine and tyrosine. Tryptophan's a little bit different. This side chain group is called an indole. It's aromatic, 2, 4, 6, 8, 10 pi electrons. It's got this hydrogen bond donor here that can be important. Tryptophan is more, the nitrogen makes tryptophan more electron rich than phenylalanine, so tryptophan's even better than phenylalanine at adopting or participating in cation pi interactions. All right. Next, the cations. Let's do the two easy ones, lysine and arginine. Again, lysine, we already used L for leucine, we already used Y for tyrosine, the S we're going to use for something else, so we use the letter K. And again, I'm used to that. I can't think of a mnemonic device, but the letter K is lysine. Lysine's tricky because you can remember that it's just this long chain with an amine at the end, but you got to remember how many atoms are in that chain. So what I remember is epsilon. Lysine has an epsilon amino group, so you count from the backbone, alpha, beta, gamma, delta, epsilon. And then you've got the amino group, which the ammonium conjugate acid has a pKa of 11, so 99% of the time this lysine is positively charged, and that's important in a lot of different situations for ion pairing. Lysine's quite happy in helices, a little less so with sheets. Lysine is the first one we've seen that has a substantial amount of left-handed alpha conformation. Another amino acid that you've seen on your previous exam is arginine, which we give the letter R, which is kind of fun and allows us to introduce another one of our OChem nerd jokes, which is, what is the pirate's favorite amino acid? And the answer is arginine. Yeah, I agree. It's hardly even worth a chuckle. And so the functional group on arginine is called a guanidinium group. It's different from an amine. This is not like a bunch of stuff with 2 different or 3 different amines. This is its own functional group. It's positively charged, and that positive charge is delocalized on all 3 of the nitrogens via resonance. This acid form that's positively charged has a pKa of 12, so it's almost always positively charged. And you can see that arginine sort of looks like lysine, only we only get to the delta carbon before we have a nitrogen. So instead of with lysine, we get out to the epsilon carbon and there's a nitrogen. For arginine, we get out to the delta carbon, and then there's this guanidinium functional group. But if you look at their dihedral angle preferences, they're similar. In many cases, you can use these interchangeably. Right. Okay, other questions? Yeah.
This is the polar groups.
These are polar and charged, so cationic. Yeah. Lastly, histidine. Histidine's weird because it doesn't necessarily fit with the aromatic amino acids, even though it's aromatic, and it doesn't fit that well with the cationic amino acids, even though it can be positively charged. So this is the neutral form of histidine. You've got the beta carbon, and then this imidazole ring. This is what you call an aromatic 5-membered ring with 2 nitrogens bonded in this way, imidazole. And the lone pair electrons on this nitrogen, are involved in the pi system of the ring. You've got 2, 4, 6 pi electrons. However, this lone pair is not involved in the pi system of the ring. It's held in an sp2 orbital, and it can be protonated. This is the conjugate acid form, which is called an imidazolium ion, and it has a pKa of 6. What that means is that at physiological pH, histidine is mostly, maybe 90% of histidines are neutral and maybe 10% are positively charged. That's really useful in enzyme active sites. So you've seen an example back in the chapter about esters and carboxylic acid derivatives where we used histidine as a base to deprotonate a nucleophile to hydrolyze an ester bond. And the ability of histidine to switch readily between this positively charged acidic form and this neutral basic form at physiological pH makes it really, really versatile. So you're going to see histidine a lot in enzyme active sites. All right, those are the positively charged ones. Here are the negatively charged ones. These are kind of boring. They just what you would expect if you wanted to put carboxylic acid on a side chain. So for aspartate, you can call it aspartic acid, but at physiological pH it exists as its conjugate base form, which is why I'm calling it aspartate. You attach a carboxylic acid group to the beta carbon. At physiological, that pKa is around 4, so at physiological pH it's negatively charged. Aspartate is one of the weirder amino acids because it's getting more and more of this left-handed alpha helical conformation. And there's something about this carbonyl group at the gamma carbon that enables that kind of thing to happen. Aspartate's okay, pretty good in alpha helices, not so good in beta sheets, maybe okay in the polyproline type area. Glutamic acid is homologous to aspartate. It has one additional methylene group in the backbone, one more CH2 group. So instead of having the carboxylic acid group attached to the beta carbon, here it's attached to the gamma carbon. It's one carbon longer, which you'd think wouldn't make a big difference, but not having that carbonyl there seems to prevent glutamic acid from being very happy with these unusual dihedral angles. But both of these will participate in ion pairs. You will also see sometimes they are nucleophiles or acids or bases in active sites. When we did the serine lipases that hydrolyze triglycerides, aspartate was in that active site as well. Okay, so, and at this point we completely give up on letters that have anything to do with the name. So aspartate is D and glutamate is E. And you just sort of have to memorize that. All right, questions about that? Okay, the polar ones that are neutral, you have serine, which is like alanine with an OH group. Then you have threonine, which is like serine but has an extra methyl group. Threonine is our third and final beta branched amino acid, meaning that you have a branch point, and it's kind of like valine, only this is an OH group. If you compare the Ramachandran plot for threonine with that for valine, here they are similar. That is, they're both okay in alpha helices, and they're both strongly favored in beta strands. And so you'll frequently find threonine in a beta sheet. Cysteine is the sulfur version of threonine, and cysteine is weird because the sulfur can do oxidation chemistry. The sulfur is also a really good nucleophile, so you'll often find cysteines in enzyme active sites. There's a whole class of proteases called cysteine proteases, and they're using the nucleophilic cysteine side chain. But here is what can happen if you oxidize cysteine. It can make a covalent sigma bond with another cysteine sulfur. And we can talk later about how that's oxidized. But this is often a great way to make a cross-link between two different parts of a protein, and that can rigidify a protein structure and make it more stable. One thing that's useful to know is that the cytosol in eukaryotic cells is a reducing environment. So there aren't any disulfide bonds in proteins in the cytosol. Disulfide bonds form in proteins that are exported through the ER and the Golgi because the ER and the Golgi are oxidizing environments. So a lot of proteins that have disulfides are secreted or displayed on the cell surface. Antibodies, for example, have several disulfides that hold them together. If you go to make a protein, and you have multiple cysteines, getting them to adopt the correct disulfides is kind of difficult. Okay. Then the other two that are polar but neutral are the amide versions of aspartate and glutamate, so they have similar names. In asparagine, we replace the OH of aspartate with an NH2. This is now an amide functional group. But look at its dihedral angle preferences. It's basically the same as aspartate. It's happy in alpha helices, but in both right and left-handed senses. And so it appears that it's really the presence of this carbonyl on the gamma carbon that makes it possible for this to adopt unusual dihedral angles. Glutamine is the amide version of glutamic acid or glutamate, where you replace the OH of glutamate with the NH2 group. It also has a similar dihedral angle preference, as does glutamate. These side chains are great at hydrogen bonding. I don't know how you're going to remember N and Q, so good luck with that. I have to memorize it. When this was probably a try-hard thing to do, but I made up little mnemonic devices for myself, and I somehow grouped aspartate and glutamate with asparagine and glutamine, and then DENQ, I imagine saying thank you or thank you or something like that. It clearly, I still remember it 20 years later, so whatever. Glycine is the easy one, and now with glycine, you don't have a side chain at all. That opens up the whole other side of the Ramachandran plot. So glycine is comfortable in both right and left-handed alpha helices. It's comfortable in beta strands and in polyproline helices. And in particular, if you need a left-handed alpha conformation, glycine's a good choice. That happens often in turns between beta strands, and so glycine is frequently found in turns. And then finally proline, which we've already talked about, which is this side chain that bites back on the backbone nitrogen. Okay, so those are the 20 amino acids. We call these, sometimes people call these natural, but there's a lot more amino acids that occur in nature, so natural doesn't really do it. Some people call them common, but there's actually a few other amino acids that are more common than some of these, so that doesn't quite do it. Canonical is okay, but who establishes the canon and what made them so important? So I prefer proteinogenic. These are the 20 proteinogenic amino acids. These are the ones that our genetic code encodes for. I just ended a sentence in a preposition. That's okay, though, right? Years ago, it is said, one of Winston Churchill's assistants wrote a memo in which he had gone out of his way to avoid ending sentences with prepositions, and reportedly, Prime Minister Churchill responded with a note saying, "This is exactly the sort of nonsense up with which I shall not put." Sometimes it's just better to break the rule, right? Okay, so those are your amino acids. Now, what we're going to talk about now is how sequence relates to structure. And I'm going to tell you a story about probably the best understood system for the relationship between sequence and structure. So I'm going to write down an amino acid sequence. I realize that you don't have the letters memorized. Well, you should, or you should be working on that, but I'll have to talk you through this sequence. By the way, when we write amino acid sequences, this is really, really important. We always start with the N terminus and we go to the C terminus. Always. Always, 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 always. And the reason for that is, as I said, peptides and proteins have directionality. Saying I want a peptide where I have alanine followed by valine from N to C is totally different than saying I want valine followed by alanine from N to C. But if you get the N to C part mixed up, and go home and draw these and convince yourselves they're different. They're very similar, but they're structurally different. They're not the same thing. So you have to write out your sequence from N to C. That is just assumed. If you write it the other way, you won't make the protein you think you're making. So here's a sequence, a 33 amino acids sequence that adopts a structure that is understood really well. I used to have something like this memorized, but I don't anymore. VELLSKNYHLENEV ARLKKLVGER. Okay. 30 residue amino acid sequence. The C terminus of this amino acid is the carboxylate group. That's the carboxylic acid group of the C terminal amino acid. And then the N-terminus should be the positively charged ammonium group, though for reasons that we'll talk about, sometimes we modify that to get structures that we want. This is a sequence derived from a yeast transcription factor, and it's a sequence that makes what we call a leucine zipper or a coil-coil. So let's recall that the leucine side chain looks like this. Here's the alpha carbon, beta, gamma, and then you've got those two methyl groups. So leucine is a nonpolar amino acid. Other amino acids you see in this sequence that are similar are valine, and let's see, no, that's it. I don't think we have any isoleucines in this sequence. So let's go ahead and highlight in orange all the times we see a nonpolar amino acid. And I guess I should also have included methionine. All right, let's talk about all the times we see a nonpolar amino acid. There's one, there's methionine. Let's see, keep going, there's a valine, there's two leucines. Keep going, there's a leucine, there's a valine, there's a leucine, there's a leucine, and a valine. Alanine is, yeah, does alanine count? It is nonpolar, but it's way less nonpolar than the others. And so we won't consider it to be, yeah, we won't think of it as being as nonpolar as the other ones. Okay, so let's talk about nonpolar side chains. Oil and water don't mix, right? And you probably learned the reason for that in 351. To summarize, we could talk about this in a lot of detail, but I don't. Hopefully, we won't need to. To summarize, when you have a nonpolar functional group that's exposed to water, the water has to be around that nonpolar functional group, but it can't interact with it through hydrogen bonding. And that nonpolar group takes up space that other water molecules could have been in, and they would have interacted with the water molecules that now surround the side chain. Basically, what happens is you get sort of this rigid structure of water molecules that surround nonpolar functional groups. It's almost like a small coat of ice around nonpolar functional groups in water. And that lowers the entropy of water. You had water molecules that used to be happy floating around in the bulk, swimming around with lots of entropy, whee, and now they are stuck. And that's supposed to be a frown. I think it, yeah. I miss my entropy. And so it's unfavorable to have nonpolar groups exposed to water because you force water to be ordered around those nonpolar groups. So one thing you can do is sequester those nonpolar groups from water, have them interact with each other, and then that releases water molecules that used to be surrounding the nonpolar groups back to be happy with lots of entropy. So imagine a peptide where you have, or protein where you have lots of different nonpolar groups, and in the unfolded shape, these are surrounded by ordered water molecules. But if you do what's called folding, if you have the backbone of the protein fold back on itself, you can create a situation where those nonpolar side chains are in the interior where they're away from water and the water is free and happy. We shouldn't anthropomorphize molecules. They don't know anything. They're neither happy nor sad. But this is what's called the hydrophobic effect. So burying nonpolar side chains away from water is called the hydrophobic effect, and that is a major driving force for protein folding. Now, if we look at this sequence, we can ask ourselves what kind of structure might allow the nonpolar side chains to be buried away from water, and can we predict it? So I'm going to point out a pattern in these nonpolar side chains that's kind of difficult to recognize if you don't know about it, but once you know about it, it's easy to find all over the place. And like any pattern in biology, there's going to be exceptions along the way. But I'm going to start with the letter A on this methionine nonpolar residue, and then I'm going to go A, B, C, D, E, F, G. And then I'm going to notice that by the time I get to G, I found another nonpolar residue. So I'm going to try again, A, B, C, D, E, F, G. And one of the things I will notice is that in these first 14 amino acids, I have nonpolar side chains at the F and the D positions. Now there is one here at E, but the overall pattern is A and D are nonpolar. You see that? It's like every 3 amino acids and then every 4 after that, you've got a nonpolar side chain. So we're going to call this repeating sequence a heptad because there are 7 of them. And if you continue on, A, B, C, D, E, F, G, there's a weird one here. That polar amino acid is actually doing something special, which we don't need to worry about right now. But again, D is a nonpolar amino acid. Then again, A, B, C, D, E, F, G. The pattern is fulfilled, then we get one final A. Again, there are exceptions. There are a couple of places where nonpolar side chains are at positions other than A and D, and there is one A position that has a polar side chain. But every 7 amino acids, we're repeating back to a nonpolar side chain at the A and D positions. Now, what kind of structure might allow those all to be on the inside of a protein? Well, let's look at the alpha helix. The alpha helix, we've talked about the dihedral angles. One of the interesting patterns in an alpha helix is that a side chain, the backbone carbonyl oxygen here in red is hydrogen bonding to the backbone NH group from another amino acid. And if you count and call this the I position, I plus 1, I plus 2, I plus 3, and then this nitrogen belongs to the I plus 4 amino acid. So you've got this hydrogen bonding pattern between carbonyl oxygens at the I position and amide protons at the I plus 4 position. And on average, the alpha helix repeats. It has about 3.6 amino acids per turn. Now that is interesting because we've got 7 amino acids in this sequence. So we could almost fit, we could fit probably one of those 7 amino acid sequences within about 2 turns of a helix. And in fact, if we tighten that up to about 3.5 amino acids in a helix, then every 2 turns we've gone through a complete heptad. And what that's going to do for us is it's going to create a situation like this where the nonpolar residues at A and D are going to be on one side of the helix with polar residues on the other side. Now they're still exposed to solvent. We're going to solve that problem next time. Okay? So see you on Monday.